Picture this. You're pruning roses on a Saturday morning. The shears slip, there's a flash of pain, and suddenly a hand is gone. In the trauma bay, doctors keep you alive, but a research team asks for one tiny slice of tissue. Why? Because they're chasing a wild idea. Maybe, just maybe, that hand could grow back. Here's the thing. Comic books have never working back. 3 months, the brand win set of challenges to first way that made the best for naught. Here's the thing. Comic books have make it use for me, considering that the hands to get them word. For though, if you're pretty much more eventually, never been they grow back. Gets what you know where today we're talking about the real science that's dragging that fantasy into hospital labs. Genetic switchboard. Instead of staring at that tissue sample and shrugging, scientists dug into DNA. What flips the body's internal rebuild switch? And just as important, what tells it when to quit? To find out, they turned to nature's regeneration rock stars. Salamanders that shrug off lost legs, and tiny fish that patch shredded fins like it's no big deal. Researchers lined up salamander and fish genomes against ours, hunting not for brand new genes, but for the missing pages in our manual. Shockingly, the trail didn't lead to thousands of exotic sequences. It pointed to a compact, quiet control cluster hiding inside our own genome. What do those pages actually do? When the team zoomed in, they spotted a pattern. These sequences blazed with activity only after injury. Emergency beacons that flashed on, then faded. In animals with super healing powers, the beacons stayed lit longer and in more cell types. That timing hinted at a hidden schedule. Not just go, but how long, how fast, and where. Meet the SP family. Crack open that control cluster and you find 10 related transcription factor genes. Scientists nickname them the SP genes. Think of them as project managers. They don't pour concrete or lay bricks themselves. They hire the crews, hand out blueprints, and decide how aggressively everyone works. Testing the theory, turning genes off. To prove the SP genes weren't innocent bystanders, researchers used CRISPR to switch them off in animals that normally regenerate with ease. A single SP knockout gave weird results: shortened bones, crooked joints, soft tissue looking for a place to connect. Knock out 2 or 3 SP genes and the blueprint developed giant holes. Whole sections never formed. SP7, the star player. One gene, SP7, yelled the loudest. We already knew SP7 steers bone-forming cells. Remove it during regeneration and the process nearly collapses. Message received. The SP family isn't flipping a generic heal switch. It's directing who builds what, where, and when. Cranking them up in mice. Now for the gutsy move. Instead of silencing SP genes, what if we turbocharge them in a creature that usually heals poorly, say, a mouse? Using an AAV gene therapy vector, scientists wired mouse SP controls to regulatory elements borrowed from salamanders and fish. Goal? Nudge regrowth a bit further, not turn the mouse into a lizard. It worked, partially. Digits came back longer, bones looked more normal, and tissues found the right alignment. Still, the mouse wasn't waving a brand new paw. The experiment screamed one truth. SP genes are pivotal, but they're only one layer in a web of immune cells, blood vessels, nerve signals, and even mechanical forces from movement. Analogy time. Think of a half-renovated house. The architect, our boosted SP genes, has drawn brilliant plans. But the electricians, nerves, inspectors, immune cells, and delivery trucks, blood vessels, still need coordination. Without them, the project stalls. Next steps in real animals. Practically, that means testing SP-targeted therapies in bigger animals with slower healing, ones that put weight on complex joints and demand fine motor control. It also means local, temporary activation, short genetic pulses right at the wound, rather than flipping the switch body-wide. Engineers may pair SP tuning with bioscaffolds or smart casts that guide shape, turning crude regrowth into precision reconstruction. Let that sink in for a second. What it could mean for people. If doctors can reliably fine-tune these pathways, catastrophic injuries might be treated like complex remodels, not permanent losses. Early trials would likely start with children, little humans already flirting with large-scale renewal, and with localized injuries such as fingertips, where control is easier. But before anyone volunteers, the safest work will happen on lab chips. Picture tiny, mini limbs grown from human cells acting as flight simulators. Researchers can crash, reset, and refine protocols long before a real patient leaves the runway. Your turn. 3 questions to chew on. 1. If scientists can flip a single, holy grail switch in animals, where in your own health are you still thinking too small, maybe a nagging knee injury or half-hearted rehab plan? 2. What's one concrete step, reading the original study, following the researchers, or sketching how limb regrowth tech could change prosthetics, that moves you from sci-fi to within my lifetime thinking? 3. If limb regeneration becomes real, how might it challenge your beliefs about disability, aging, and irreversible damage? And who could you discuss that with this week? So here's what we covered today. First, the hunt for a genetic switchboard led scientists to a quiet SP gene cluster already hiding in our DNA. Second, turning those genes off mangles regeneration. Turning them up pushes it further, at least in mice. Third, SP genes act like architects, but a whole crew, immune cells, nerves, blood vessels, still has to show up for work. Fourth, the road to human therapy runs through larger animal studies, local gene activation, and lab-grown mini-limbs. The bottom line? We're closer than ever to regrowing complex body parts, but it's not one magic gene, it's a symphony, and the SP family is just the conductor. Take that thought into your week. Look at any area of recovery in your own life, physical, emotional, even professional, and ask, am I settling for scar tissue when I could be rebuilding? Until next time, keep your curiosity switched on and your blueprints handy. Who knows, your body might be capable of more than you've ever imagined.